A vaccine can protect a person against a disease-causing pathogen in a number of ways. Here we are going to see how a viral vector vaccine works. Administration of a vector vaccine provides our bodies with a genetic blueprint for the protein of a pathogen. This genetic blueprint consists of deoxyribonucleic acid, or DNA for short. The transport vehicle which carries the blueprint of a pathogen's protein into the cells of the body is called a vector. Harmless viruses, for example, can be used as vectors. For safety reasons, when being used for a vaccine, they are modified so that they continue to infect human cells but cannot make people ill. For this purpose, a piece of DNA in the vaccine vector is switched around with the DNA blueprint for a pathogen protein. After the vaccine has been administered, the vaccine vectors are the first to dock onto the cells and penetrate them. Once they get inside the cell, the genetic blueprint of the vector, its DNA, is transported into the cell nucleus. The DNA of the pathogen's protein is transcribed into messenger RNA inside the nucleus. The mRNA blueprint is transported out of the cell nucleus and docks onto a ribosome. The ribosome has the ability to read the blueprint of the mRNA. It produces the target protein according to the instructions. The vaccine contains the instructions for making the protein of a pathogen. The newly produced pathogen protein is transported to the surface of the cell so that it can be presented to the immune system for inspection. The immune system recognizes that it is a foreign protein which may belong to a dangerous pathogen and therefore needs to be eliminated. The immune system responds by unleashing a whole arsenal of defense mechanisms. Antibodies in particular, along with specific defense cells, are formed that target and eliminate the pathogen's protein. And to provide us with long-term protection, our immune system remembers which antibodies were most effective against a specific pathogen. Vector vaccines employ the principle of a harmless virus to infect the cells of the human body efficiently. The difference, though, is that a vaccine vector does not cause an infection and hence an illness. The pathogen protein which triggers the immune reaction is produced in our own cells. Vector vaccine technology has been established for many years. There is a vector vaccine to control the Ebola virus, for example. Further promising uses are currently the focus of clinical trials.